Hello and welcome. Today we're going to be reviewing a lab on cell respiration. The main purpose of this lab is to demonstrate the active process of cell respiration, which involves converting sugars and oxygen gas into water, carbon dioxide, and a usable energy, in this case ATP.
To make the experiment less complicated, we decided to use yeast as the organism on which we would observe the effects of cellular respiration. Using the standard equation for cellular respiration, we hypothesized that yeast would respirate more with a larger quantity of sugar present, and the solution with more sugar would thus produce a larger amount of carbon dioxide gas.
To put this into more specific terms, the amount of sucrose sugar would be our independent continuous variable, while the height of the carbon dioxide bubbles would be our dependent continuous variable.
Thanks to the myriad of cell respiration labs available on the internet, it wasn't hard to find a viable experiment to perform. However, to make it slightly more challenging, we selected one that incorporated a new aspect, inverting test tubes to create a sealed environment, which would limit the amount of oxygen present. With one minor exception, we performed the lab identically to the given procedure. The only aspect of the procedure changed was the amount of yeast solution. The procedure calls for 1 packet of yeast, which is roughly 7 grams, dissolved in 900 milliliters of water. This large quantity was intended for several groups to use at once, and, not possessing the large beaker or the individual packets of yeast required, we had to recalibrate our sample size. Our finished solution held 500 milliliters of water and 3.88 grams of yeast, which was proportional to the original solution, but on a much more small and manageable scale.
Our experiment required a total of two 500 milliliter beakers and 6 test tubes, 3 large and 3 medium. As previously mentioned, the smaller test tubes would be used inside the larger ones. We filled the larger test tubes with yeast solution, and then weighed out and poured 2.5 grams of sugar into one test tube and 0.2 grams into another, leaving the third test tube with only yeast solution to serve as the control, because its absence of sugar would prevent cell respiration, and it would therefore fail to produce any carbon dioxide bubbles. The smaller test tubes were then inverted and placed into the larger test tubes, already containing the yeast solution. Each sealed environment was placed into a large 500 milliliter beaker of water, kept at 30 degrees Celsius, the appropriate temperature for yeast to undergo respiration at a delayed and observable rate. The second larger beaker was merely used to hold the yeast solution. The test tubes were left at this warmer climate for 5-minute intervals, for a total of 45 minutes. At every 5-minute interval, the test tubes were removed and the bubble production was observed and recorded in our data table via ruler measurements in centimeters. At the end of the 45-minute trial period, the test tubes were appropriately cleaned out and the remaining yeast solutions were disposed of accordingly.
From the data obtained in this lab, we were able to clearly see the trend displayed here. A larger amount of sucrose, which in this case is the limiting reagent, produced more carbon dioxide gas. A smaller amount of sugar resulted in the production of less carbon dioxide, and the control did not produce any because there was no sugar present to react with the oxygen.
According to the graph, which measures the relationship between time elapsed and carbon dioxide bubble height, the solution with 0.2 grams of sucrose produced carbon dioxide bubbles that reached 1.5 centimeters after 15 minutes and remained at that level due to the fact that all of the sucrose present had respirated. In the case of the 2.5 gram solution, the bubbles reached a height of 8 centimeters at 35 minutes, which was the maximum height within the smaller test tube, which therefore made it difficult to determine whether the completely respirated sucrose produced that height of bubbles or a greater one. Nonetheless, there is a linear relationship between the two variables.
Overall, our hypothesis was fully supported by the data, as there was a direct relationship between the amount of sucrose present and the production of carbon dioxide gas. We have in fact verified that there is a direct relationship between the amount of sugar present and the amount of CO2 gas produced in a respiration equation.
Thanks for listening. We hope you enjoyed it.